Okay, well, thank you for having me. So first of all, for to be honest, I didn't start my science try to understand or treat diabetes, and then my research in the early days was to try to really fundamentally understand bioenergetics. And brown fat is simply a model because brown fat contains a lot of mitochondria. In the early days, I thought probably comparing a lot of brown fat, like a lot of mitochondria, versus white fat, have very little mitochondria. Maybe comparing those cells may tell us about mitochondrial bioenergetics. That was the original idea. And then I didn't really intend to study diabetes. The reason we got into this field was just really an unexpected observation. Early days, I told you, we tried to understand transcriptional pathways, how brown fat development is transcription regulated, and eventually led to the discovery of this complex called PRDM16 and its subsequent sort of the associated factors like EHMT1, PGC1 alpha, etc. This actually allow us to make the first mouse model that lacked brown fat and the beige fat for the first time without using diphtheria toxin. Back in those days, 90s, people made brown fat ablation using diphtheria toxin, but it has some caveat because diphtheria toxin has a lot of inflammation. So around the time when I start my lab, we made first mouse model that lack brown fat. And then initial anticipation for us is all about thermoregulation. We thought about regulation, but it turns out, surprisingly, the mice that lack brown fat have severe insulin resistance and type 2 diabetes, and which I did not anticipate. But it was really puzzling because that time brown fat is about all about thermogenesis by mitochondrial uncoupling protein one called UCP1, and then UCP1 knockout mouse are not diabetic at that time. However, when we made a mouse that lack brown fat, they instantly become diabetes. So we're really puzzled. What does it even mean? And a lot of pushback at that time. But eventually that led us to think of the idea that maybe brown fat is doing something more than making heat by UCP1. Then later, we did a converse experiment to show that brown fat, beige fat doing something completely different from UCP1. And eventually that led to the discovery of UCP1 independent pathway. So it's a long journey. And then that's what I said, unexpected. I didn't anticipate to study diabetes, but that's science released us. Well, it's a great question and still a lot of discussion around this. But just I want to put us in the same page. So the definition of brown versus beige. Okay, so the brown fat initially was really about embryonic form of brown adipocytes, which is very developmentally fixed adipocytes that give rise to thermogenic adipocytes in mice, in particularly intrascapular regions or infants. And it's a very developmentally fixed process. On the other hand, beige is when we were at Bruce Spiegelman's lab, we start to call them beige because those inducible brown adipocytes are surrounded by white adipocytes, and as a tissue level, it looks less brown, right? So that's why the color implicates sort of different inducibility and also different developmental lineage, and eventually that led to the notion that a beige is more inducible form in adults, and then brown adipocytes are more embryonic form. Now, human brown adipose tissue, right? BAT, brown adipose tissue contains different cell types, right? Endothelial cells, adipocytes, neurons, many others. Within that tissue, we're fairly convinced that human brown adipose tissue is inducible. For multiple investigators, not only us, show that the brown adipose tissue in humans contain very beige-like inducible form of thermogenic cells exist, and then chronic activation of this tissue by chronic cold activation or chronic beta 3 receptor agonist or certain kinds of disease chronically form or recruit new form of brown adipose tissue, even for those who have lost active brown adipose tissue. So in that regards, probably brown versus beige a little confusing, but maybe we could say developmentally fixed form versus highly inducible form. That's sort of the definition of brown versus beige. Now, your question is about how we evaluate that, right? So far, only way to evaluate brown adipose tissue in human is FDG PET scan. It's not realistic because you need isotope, expensive instruments, and highly inducible by cold. So we, and as a field, need biomarkers, right? Probably that recapitulate the amounts of thermogenic fat cells. That's where we are now. Yes. So maybe your question is about what is the emerging debate or question, right, in this field. I think I would think a couple of things, but first, probably the most important area we as a field have to resolve is that is really the brown fat in humans therapeutically targetable to achieve certain clinical benefits, right? We know that humans, healthy adult humans, do have active brown adipose tissue. We do know that the amount or prevalence of brown adipose tissue really significantly correlate with reduced odds ratio of type 2 diabetes, means that a brown fat is very cardio-metabolically healthy, good fat, right? Now, what we still don't fully understand or we don't know is that why brown fat is a good fat. We assume, or as a long-standing consensus for many years, is that it's about thermogenesis. But we just described, don't really think that thermogenesis is a driver. It's far more than making heat. Brown fat is clearly doing more than making heat. And exactly what aspect of this function related to the metabolic benefit? That's a one thing. The second is about really, if so, then can we target that aspect to improve metabolic health as a therapeutic target? These are probably the major question as a field. It takes all village to figure that out for the next 5 years or so. Yeah, it's a great question. And so, first of all, I'm not in that race of or debating in particularly beta 2 versus beta 3. So I'm going to give you a little more neutral, unbiased view. I think the beta 3 idea was very compelling by Aaron Cypis and others, clearly show that beta 3 receptor is expressed not only in the mouse but also human brown adipocytes or beige adipocytes, and activation of that can be therapeutically activating brown fat. On the other hand, two groups actually show the beta 2 is also expressed and then have some compelling evidence, means suggesting that's the case. I can't really tell which one is right, but this also, again, back to this debate, that really is the opportunity to resolve. I consider this is a great opportunity to understand better, and hopefully, maybe both are right. That we don't know. Back to the next question around like which part regulates UCP1 versus UCP independent. At least in calcium cycling, we know that activation norepinephrine signal activate alpha 1, which trigger calcium cycling, as well as beta 2, beta 3, which also activates cyclic AMP downstream signal. So in that sense, then both, if I can speak about UCP independent calcium cycling, I think beta 2 or beta 3 both can activate UCP independent pathway.